Chapter 15, DNA and the Gene. In this chapter, we're going to be talking about a specific phase of the cell cycle. And this cell cycle is the S phase. This is where DNA is synthesized. Another way of saying it is making a copy of DNA. In this chapter, you will learn DNA replication and repair to preserve genetic information. We'll talk about what genes are made of, DNA synthesis, the early hypothesis, steps in replication, and then lastly, we'll talk about telomeres, the end of linear chromosomes, and we'll also talk about ways to correct mistakes made during copying of the DNA molecule. So experiments have led to discoveries of classical genetics. Remember, when Mendel was looking at his pea plants, he had no idea of a chromosome or the genes located on the chromosome that code for traits. The molecular nature of genes and how they were copied remained a mystery for a long time until maybe the 1950s. Studies identified the nature of genetic material and how genes are copied during synthesis. And so this is what this chapter is about. So chromosomes are in all living cells and they carry information. And remember, information is one of the 5 critical characteristics of life itself. So all genes are made of DNA and they're not made of protein. But how do we know this? There was a very classic experiment. And this is how we determined that DNA is in fact the information molecule. So in this experiment carried out by Hershey and Chase, what they did is they looked at a common bacteria found in the guts of most mammals. And this bacteria is called Escherichia coli. They knew that there was a virus that infected it called T2. So the question is what is injected by the virus? So if we step back a little bit and think, what is a virus composed of? We can say that a virus is actually made of basically 2 things. It's made of a capsid or a protein coat, and in the inside it has a piece of genetic material that can be either DNA or RNA. So what is it that was injected into E. coli by the T2 virus? So what they did is they labeled certain minerals that are found only in the information molecule. Phosphorus is only found in DNA, and sulfur is only found in proteins. So, excuse me, if the labeled sulfur was found inside of the E. coli cell, then we know that proteins are the information molecule. On the other hand, if labeled phosphorus was found inside, then we know that actually DNA is the information molecule. Okay, so here we see labeled phosphorus and sulfur for DNA and proteins respectively. And so what is it that the scientists found inside the cell? And you already know the answer. They actually found DNA. So that means they found labeled phosphorus. And this proved that DNA carried information to make more viral particles. And so here you see the setup of the experiment, and you can read more in your books. So let's talk about the structure of DNA. And when you think about this, this could be a review from what you had earlier in this semester, maybe chapter 3 or 4. So it's reminding us that DNA is double-stranded. It's the double helix. And each nucleotide, or the proper term, deoxyribonucleotide, is composed of sugar, phosphate, and a nitrogenous base. And each of these nucleotides are linked together by a phosphodiester linkage. And remember, this is a type of covalent bond. So we notice here that there is directionality to DNA. One side ends in the 3' hydroxyl group and the other side ends in the phosphate 5 prime. Now remember what 3 prime and 5 prime is? You're right, it's the carbon. It's the carbon found in deoxyribose. Remember, it's 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. It's all the way up to 5 because deoxyribose is a pentose, a 5 carbon sugar. The structure of DNA, remember the backbone is alternating sugars and phosphate. So sugar, phosphate, sugar, phosphate. So it's sugar and phosphate. And then the steps to the ladder are these bases: apple tree chewing gum, adenine, thymine, cytosine, and guanine. And remember that there's complementary base pairing. A always complements T and C always complements G. I love figure 15.3 because on the left-hand side, it shows the 3 parts of a basic nucleotide: a phosphate, a sugar, and a base. And notice the base can be A, T, G, or C. Notice that they have only labeled 3 of the 5 carbons in the sugar. But notice, starting from one corner and going clockwise, 1 to every corner of that ring is a carbon. So we have the 3 prime and 5 prime, which is so important because it indicates directionality. And then on the other side, you see the phosphodiester linkage. You see that it is a phosphate, covalent bonds with phosphate that are linking one nucleotide to another. And notice one end of the strand ends in 5 prime and the other strand ends in a 3 prime carbon. And this gives directionality to the DNA molecule. Each strand of DNA has directionality, as I mentioned before. One side ends in the 3 prime carbon end and the other end ends in the 5 prime carbon end. And so when we take a look at the 2 strands, we know that they are anti-parallel. One strand is going in one direction and the other strand is going in the other. So they are anti-parallel strands. And these twist around to form the double helix. This figure 15.4 is very representative of what we've just been discussing. Notice it shows on the left-hand side, we see that we have anti-parallel strands, and we can detect the directionality by 5 prime, 3 prime. Notice that they're at opposite ends and the strands are anti-parallel. The other thing that you should notice is that there is complementary base pairing. A always pairs with T and C always pairs with G. And then it forms a double helix. And this has a lot to do with the hydrophobic, hydrophilic nature of the DNA molecule. Anti-parallel strands twist to form the double helix, and we see complementary base pairing. So G always bonds with C, and A always bonds with T. So that's complementary base pairing. So it says here that the double helical DNA molecule is stabilized in 2 ways. One way that it makes it a very stable molecule is, in fact, the complementary base pairing. And the other way is the interaction between stacked bases inside the helix. So it is a very stable molecule, and when you think about it, stability means that it's not chemically reactive. Stability means that it is going to keep your information. And isn't that what we want? Isn't that the function of the molecule? Is to make sure that it isn't reactive, that it won't change, and that it will maintain its integrity. So there is an early testing of a hypothesis about DNA synthesis. And so there were 3 ideas that were being tossed around. How is it that DNA replicated during the S phase of the cell cycle? Remember, during interphase we have G1, S, and G2? Well, during the S phase, is it semi-conservative replication, 1 strand old, 1 strand new? Is it conservative, 2 strands old, 2 strands new? Or is it dispersive, little bits and pieces of old and new strands like randomly combined? Which way was it that DNA replicated? I'm going to sort of cut to the chase and just tell you that it is semi-conservative replication. Okay, so what is happening in semi-conservative replication? Semi-conservative replication means that the parent strands unwind and they unzip. And each strand is used as a template, and it's going to build a new strand. So then the two daughter strands are one strand old, one strand new. One strand old, one strand new. And so this is semi-conservative. Conservative means that you have two old strands, and then the new molecule that's replicated is definitely two new strands. And as I said before, dispersive is bits and pieces of old and new strands. So how do we know that it's semi-conservative replication? Well, there's another classic experiment by Messelson and Stahl. And again, they're going to use E. coli as a model. So what they're doing is they're taking a look at the DNA inside of E. coli. And they're going to introduce heavy nitrogen. So regular nitrogen is N14. That means it has 14 neutrons. Excuse me, 7 protons and 7 neutrons in the nucleus. But N15 has 7 protons and 8 neutrons. So it is heavier. So we can test to see exactly what happens if we weigh the DNA molecules if we have introduced a heavy form of nitrogen. So this is how this test was actually made. First of all, the chromosome, the circular chromosome, was actually extracted from E. coli. And then you can see in red up at the top in the Erlenmeyer flask that N15 was introduced, heavy nitrogen. And this heavy nitrogen was introduced in generations 2 and 3. So the following diagram below actually shows what happens if you buy into semi-conservative, conservative, or dispersive replication. So in actually semi-conservative replication, notice that there are two strands that are entirely made of N15 and then two other strands that are 50% N14 and two that are N15. So we should get a weight in between. Then if you see conservative replication, you should see one strand that is entirely made of N14, but 3 strands that are made of N15. And then lastly, if you look at the second generation with dispersive, you see that bits and pieces form old and new strands. So the truth is what they did is they found out that there were actually two when they did electrophoresis, there were 2 bands, and that matches up with semi-conservative replication. So this is how we know that DNA forms, and the type of replication is called semi-conservative replication. Okay, now what helps add the nucleotides? We have one old strand and here's the new strand, but how do we build the new strand? Well, there's this enzyme called DNA polymerase. It's like my hand, and it's going to start adding the nucleotides as we build it. So my hand is DNA polymerase. So DNA polymerase is an enzyme that catalyzes DNA synthesis. There are several types of DNA polymerases, and we'll talk about them in just a minute. And it says here, the thing about DNA polymerase, it can only add nucleotides to the 3' end of a growing DNA chain. So therefore, it can only move in the 5' to 3' direction. So what does this mean? Okay, like for me, I'm going to simplify it, and I'm going to just say to myself, this reminds me of when I was a young girl, ages and ages and ages ago, is that when we had toys and they moved, they were usually wind-up toys. Like you would wind and crank the key, and then you would see like a little duck or a robot, start walking across, and when it hit the wall, it would just continue going like this. It was stuck because it could only move in one direction. But nowadays, with the holidays coming up, maybe some of you are going to get a drone for a gift. A drone goes frontwards, backwards, sideways, from left to right, and then it goes up and down. So it doesn't, so DNA polymerase does not have that kind of movement. It only has the movement of an old-fashioned wind-up toy, like a toy that I would get. So it can only move in the 5 to 3' direction because it can only add on to the 3' end of a DNA chain. Okay, so DNA polymerization is endergonic, and I know that you remember what endergonic means. It means that it requires energy to build a large molecule, and that makes sense because where's all the energy stored? In the bonds, right? And so if energy is stored in the bonds and you're making more bonds in a larger molecule, then it needs energy. But this energy comes from activated nucleotides. And I hope that you'll remember it from chapter 4. So activated nucleotides are called deoxyribonucleoside triphosphates. In other words, that triphosphate tail of the activated nucleotide provides that energy to actually create the phosphodiester linkage. 2 phosphate groups break or cleave, and 1 remains, and this is how we add 1 nucleotide to another. So it is true, DNA synthesis does require energy, but that energy comes from the building blocks themselves because they're not just nucleotides, they're activated nucleotides called nucleosides. In this diagram, figure 15.6, you can see on the left-hand side at the bottom an activated nucleotide. How do I know that it's activated? Because I see a triphosphate tail. And notice we know that 2 of the phosphate groups will break off and the remaining phosphate will form the phosphodiester linkage. So those 2 phosphates, when the bond breaks, that will provide the energy to form the phosphodiester linkage. This is an excellent diagram. You know what I also notice in this diagram with the anti-parallel strands is that notice deoxyribose is facing up on one strand but facing down on the other. This also gives you a clue of anti-parallel strands and that DNA has directionality. So where does replication start? If we're dealing with a prokaryote, remember prokaryotes don't have a nuclear membrane. So it's out in the nucleoid region is this one circular chromosome, and it's like a ring, right? When you're married, you get a ring. It's a beginning with no end because once you start, you end up in the same place that you started. So with the circular chromosome, you begin replication at the origin of replication and you end in the same place because the chromosome is circular. But then you get to eukaryotes, which have a nuclear membrane, and they have linear strands of chromosome. Like, for example, us. Don't we have 46 linear chromosomes? And so you start in one place when you start making a copy of DNA, but you end in the second place. And that is the difference between the two. This is a wonderful diagram to indicate this concept in figure 15.7. Up at the top where it says B, you have a bacterial chromosome, and notice that it's circular. And there's the origin of replication. So notice that there's this replication bubble, and it keeps on opening and opening, getting broader and broader. And within that bubble, you see that 2 strands are being made, one moving in one direction and the other one moving in the other. Why is that? That's so strange. Well, it's because DNA polymerase can only move in one direction. It can only move in the 5 to 3' direction. It can only add nucleotides to the 3' end. So notice in one strand, it's moving towards the replication fork, and in the other strand, it's moving away. So you see the new strands of DNA indicated by the color red moving in opposite directions. Now let's go to part C. Part C, we see a linear chromosome, and now we have multiple replication bubbles. But within each replication bubble, we see that new strands of DNA are being replicated, but notice that they're moving in opposite directions, and that makes sense. The two things that make the new strand move in opposite directions are this. DNA is anti-parallel, and that's indicated by the gray. The gray represents the old strands. The new strands that are being formed can only form in the 5 to 3' direction because DNA polymerase can only move in the 5 to 3' direction. So how is it that we can stabilize these replication bubbles? Well, I guess you've imagined right now that DNA polymerase does not work alone. So there are other enzymes and proteins helping out with the synthesis, because keep in mind, DNA molecule is a very stable molecule, so it needs a lot of help to catalyze reactions. So one of the first helpers is DNA helicase. DNA helicase is like a zipper. It's going to unzip the double strands. So they're together, and helicase will act like a zipper and unzip it. Then we have single strand binding proteins. Well, once it's unzipped, we don't want the strands to go together again. We want them to move apart. And so single strand binding proteins keep the strands apart, single strand binding proteins. And then we have this last one here on this slide. It's called topoisomerase. Topoisomerase takes out the kinks. So let's say I have this rubber band, and remember it's the double helix, but if I open the replication bubble, you see a lot of kinks towards my finger. How do you get rid of all those like windy kinks? Well, we have topoisomerase. Topoisomerase cuts DNA, it takes out all of the kinks, and then reconnects the molecule again. Another place where you can have a lot of kinks is I have a cord connected to my blow dryer. And when I use my blow dryer a lot, I notice that the cord becomes really twisty. So to relieve all that tension, I'll grab the cord and I'll let my blow dryer hang, and then it starts to spin and take out the kinks. That's topoisomerase. Then it says here we have two kinds of strands, because keep in mind the strands are anti-parallel, and you notice that the new daughter strands indicated by red in the previous figures in red were moving in opposite direction. So the one strand that we're going to talk about first is the leading strand. The leading strand is made by DNA polymerase in the 5 to 3' direction. In other words, it just goes like this, whoosh. It just goes in one direction, no problems whatsoever. So the reason is it's copying the, what's the opposite to 5 to 3? 3 to 5. So the old strand is 3 to 5, and the new one that's being built is 5 to 3. So it says here, DNA polymerase can only move in the 5 to 3' direction. And DNA polymerase cannot start synthesis from just the very beginning. So it needs some help. So now we're going to talk about something else. So there's still more enzymes that help with the copying of this. So this connection depends on actually having a little piece of RNA. So the primer RNA provides that little connection there, like a raised Lego peg, so that nucleotides can start connecting together. Okay, so DNA polymerase adds these activated nucleotides to the 3' end, and since both strands are anti-parallel, we have a leading strand, and another name for leading strand is continuous, because remember I mentioned once we have that primer, DNA polymerase just adds one nucleotide after another, so it just goes straight through. It zooms through in a continuous fashion, and therefore the leading strand is also called the continuous strand, because it's building the daughter strand in the 5 to 3' direction. Figure 15.8 is a marvelous indication of all of the enzymes used or needed to synthesize a new strand of DNA. So notice I see primase in yellow, topoisomerase upstream in green to take out the kinks. Then I see helicase, which is the zipper, and the single strand binding proteins to keep actually the molecule open. And then if I look at the second part over there, I see a new strand, a daughter strand being made, and I can tell because the old strand is in gray and the new daughter strand is in red. But notice at the very beginning there's a primer, and that primer is RNA, and remember it's put on by primase. So here you see DNA polymerase moving through, and directly behind it is what they call a sliding clamp. So as the replication bubble starts to open and move across, so does the replication of the leading strand or the continuous strand. Now let's talk about the lagging strand. And I know that this is difficult, but I want you to remember DNA polymerase, which is the major enzyme that puts one nucleotide, the complementary base pair on the other side, can only move in the 5 to 3' direction. So the other strand, the other old strand is already in the 5 to 3' direction. So that means the opposite of that is 3 to 5. But DNA polymerase can't move in the 3 to 5' direction. So what's going to happen? That means that DNA polymerase is going to work backwards. It's like doing a blind stitch. You go up and then you go down, and then let me start over here. You go up and down, and then you move up and down, up and then go backwards. So DNA polymerase is actually moving backwards and then forwards, backwards and forwards, backwards and forwards. And you're saying, oh boy, this is confusing. Well, it's causing a lot of fragments, and that is why another name for the lagging strand is the discontinuous strand, because DNA polymerase has to move backwards and then forwards because it can only move in the 5 to 3' direction. And so what it's causing is the formation of a lot of little fragments. So at the very top, we see time 1. We see we're at the replication bubble. So notice one strand is moving towards the replication bubble. That's called the leading strand or the continuous strand. And the bottom strand is actually being built away from the replication bubble. In other words, DNA polymerase has to move backwards and then move forwards, move backwards and then forwards. So notice that little piece is made. And then if you look at time 2 down below, then you see the replication bubble has opened even more. And so DNA polymerase has to move back and then move forward, creating a second segment. So notice that the lagging strand is discontinuous. It's made of little tiny fragments because DNA polymerase can only move in the 5 to 3' direction. Okay, so what are these little fragments called? They're called Okazaki fragments. And these Okazaki fragments, they were discovered by Dr. Okazaki, but these have to be put together. So these fragments will soon be linked together after they are formed. And this is why, again, that the lagging strand is also called the discontinuous strand. I love this frame right here because this one focuses on the lagging strand. So notice if you look at the old strand, which is indicated by the color gray, it is in the 5 to 3' direction. So if this is going from 5 to 3, then the other strand has to go from 3 to 5. Well, that's hunky-dory, but DNA polymerase can only move in the 5 to 3' direction. So what's happening is that DNA polymerase must move backwards and forwards, like an old-fashioned blind stitch when you're sewing, so that it can make a strand or a copy. So here you see at several points what is happening, and you see here this is introducing us to something else. So now we see in the lagging strand that we have a lot of little fragments here. And notice each of the fragments start with a primer, which is a piece of RNA which is put on by primase. But notice if you look towards the bottom in step 4 and 5, we see for the first time DNA polymerase 1. Remember, there's different types of polymerases, and this one is actually knocking out the RNA primer and replacing those bases with DNA bases instead of replacing them with deoxyribonucleotides. And then notice in frame 5, we see that there's something else. There's another enzyme, which is called DNA ligase, and that is connecting those fragments together. So there's two new parts, DNA polymerase 1 and DNA ligase. So if you've taken chemistry before, they talk about ligands, chemical ligands. Ligands are those molecules that can connect. And I also think in Spanish, when we think of liga, like a rubber band, it's also a type of connector too. So DNA ligase connects the Okazaki fragments together. Figure 15.10 is a wonderful illustration of Okazaki fragments. Table 15.1 is a summary of all the new terms that we've learned and all the enzymes that help synthesize DNA. And this synthesis occurs during the S phase of the cell cycle. So notice here we have all different kinds of molecules that are helping with the replication of the DNA molecule. DNA polymerase 1 removes RNA primers and replaces it with DNA like we saw in the previous figure. And DNA ligase actually forms this phosphodiester linkage between Okazaki fragments. Now, when you put all of these together, when you put all of the enzymes together, notice we have DNA polymerase 3 actually putting on the nucleotides at the beginning, DNA polymerase 1 knocking out the primer. But we also see helicase, primase, DNA ligase, single-stranded binding proteins, and topoisomerase. When you put all of these together, this is called a replisome, and a replisome is a DNA synthesizing machine. You'll be able to see the short clip of this of DNA synthesis in your other presentation that is posted in Blackboard. Okay, so what is at the replisome? So the replisome contains all of the enzymes responsible for DNA synthesis. I'm just reminding you that you do need to know all of these enzymes for your upcoming final test. And all of these are joined together to form one macromolecular machine called the replisome. Okay, so the replisome is dynamic, and it says here the continuous strand may not be synthesized continuously as once believed. So maybe there are some little hiccups along the way. But all of these together both build daughter strands from the old strand for both leading and lagging strands. Now, there is an issue because we have linear chromosomes, and that's the end of the chromosomes. So what is at the end of the chromosomes? It's called the telomere. So the ends of linear chromosomes are called telomeres. And these telomeres can be very problematic, and the reason is there's no problem with the leading strand, because the leading strand starts and it just goes continuously all the way to the end. Excuse me, but there is a problem with the lagging strand. In the lagging strand, you start at one place, and then you have to go backwards and move forward, backwards and move forward, backwards and move forward, and then you go backwards and you fall off the chromosome. So then that last little piece can't be copied. So when you're moving backwards and forwards, there is a problem on the lagging strand for linear chromosomes. So primase adds RNA primer close to the end of the chromosome, but if we're at the very end, then there's no place to add it. So DNA polymerase cannot be added without the primer, and it's almost like the enzyme falls off of the old parent strand. So single-stranded DNA is left at the end of the lagging strand. So that single-stranded DNA is degraded. So what's happening is every single time your cells replicate, your chromosomes, according to this, get shorter and shorter, and it's because of the lagging strand. The lagging strand, you can't do that last little piece at the end because you sort of, if you have to go backwards and then forward, backwards and forward, that last little piece, when you go backwards, you fall off the strand and you can't move forward. Okay, so the other strand, that last little piece that's flapping there with no partner on the other side, eventually degrades. So what this means is that chromosomes get shorter and shorter and shorter with every replication. Now what happens is, if this were to continue going, it's implying that over time linear chromosomes would vanish. But telomeres do not contain genes, and actually the ends can be rebuilt by an enzyme called, believe it or not, telomerase. So telomeres are short repeating stretches of bases, and they can be rebuilt and re-lengthened with an enzyme called telomerase. So in figure 15.12, you can see that last little bit. If we go to the very bottom of the diagram in step 4, we see that last little bit on the lagging strand that cannot be replicated. Because remember, DNA polymerase has to go backwards and then move forward. And when it goes backward, it actually falls sort of off the old molecule. It has nothing to latch onto. So then that last little piece isn't replicated. So this is what it looks like, our chromosomes. So that means, so every single time our chromosomes replicate, we see that that last little piece isn't replicated. Well, telomerase solves the end replication problem. Telomerase re-lengthens the ends of the chromosome using RNA as a template. So if you had like a scaffold, you could have this template, and this template will rebuild that end that could not be copied. So it says telomerase binds to the overhang. It's sort of like as if you had a crane and you're holding a piece of RNA, and it's taking it out to the very end of the chromosome, and it's going to use this as a template to rebuild DNA at the bottom. So instead of using my fingers, figure 15.3 shows exactly what's happening. In green, you see the enzyme telomerase. And using RNA in yellow as a template, it is rebuilding the end of a chromosome. So notice in red, nucleotides are coming in, and they're nonsensical repeats, but they're re-lengthening the chromosome end. Now, the problem, though, is that telomerase is basically found in gametes and stem cells. It is not found in our somatic cells. And I, for one, know that that's true because as an older, mature person, I notice that my skin is thinning out, and it's not nice and pretty like young people's skin, like my students. So what's going on there? Well, my cells can only replicate so many times, my body cells, my somatic cells. And so they have progressively shortened over time because basically telomerase is not found in my body cells. So a scientist, Grider, proposed that the number of cell divisions is limited by the initial length of the cell's telomeres. So once the telomeres get really shortened and almost vanish, then the cell stops to divide. And this is true also. You could take some of my skin cells, and they only have a limited number of cell divisions. Adding telomerase to cells in culture allows them to continue dividing longer. Now, I would go out and buy a jar of telomerase cream so I could have my skin cells divide longer and longer, and I'd have young, youthful skin. But the truth is that the presence of telomerase in somatic cells is also a red flag for cancer cells. So cancer cells have a lot of telomerase. And remember, cancer is those cells that are not obeying the cell cycle. They just continuously are replicating. And part of that could be telomerase. The last part of this chapter is talking about repairing mistakes and DNA damage. So let's talk about that. As a molecule, DNA polymerase, the enzyme that actually is adding the nucleotides in the proper order using complementary base pairing, is very, very accurate, but it does make mistakes. And some of these mistakes can be very serious because it can be the onset of cancer. So it's very important that the copying and the final product of DNA is as perfect as can be. So how is it that we can correct mistakes in DNA synthesis? Actually, when DNA is copied, there's one mistake in every 100,000 bases. But the truth is in the end, we have like one mistake in a billion. So how do we go from 1 in 100,000 to 1 in a billion? Well, there's measures that corrects these mistakes at the cellular level. So there's repair enzymes that remove defective bases and replace them with the correct ones. So the first form of correction is DNA polymerase proofreading. So DNA polymerase also proofreads its work. In other words, it's like when I text or type something, I have to go over it and correct my mistakes, all the misspelled words, which is all the time, by the way, guys. So it says here, DNA polymerase will add nucleotides only if the previous base pair is corrected. So there's this site, it's called the exonuclease active sites, and mismatched nucleotides are removed and the correct ones are replaced. So they remove the incorrect base pairs and then they replace them with the correct ones. So here you see DNA polymerase proofreading. So here you see at the polymerase active site, and here you see that A and C do not match each other. It should be A and T. So notice the C is knocked out, that base is knocked out, and it's replaced with the correct one in the proofreading system. There's also this thing called mismatch repair. In the mismatch repair, oftentimes DNA does proofread, but it might leave some mismatch pairs behind. And so we have a second system that comes back and takes those mismatched nucleotides and corrects them and puts the corrected base with the proper base. So we have a mismatch repair enzyme system. So this is after proofreading. So I don't know if you're aware of this, but they actually use UV light in wastewater treatment plants to actually kill bacteria. And we know that UV light does damage DNA. So how does it cause the damage? What it's going to do, it's going to cause these things called dimers. And anytime you see di means two. So two thymines on the same strand will actually start to bind to each other, causing a kink, and this is not good. So there has to be a repair system to fix this UV damage. So here we see UV light damage coming in, and we see two thymines on the same strand, and instead of binding to another base on the opposite strand, they're binding to each other, forming a kink. The nucleotide excision repair system recognizes these kinks. It takes out the kink and replaces it with nucleotides that are not damaged. And then DNA ligase links the repair strands to the original undamaged DNA. So here you see the nucleotide excision repair system. You see two thymines that are actually binding to each other instead of to adenine on the opposite strand. So the excision repair system removes those bases, the damaged bases, and replaces those with those that are undamaged. And now at the very last step, in step 5, you see that everything is perfectly matching. So I'm not talking about an albino, but I'm talking about a person that does not have the excision repair system. In other words, we're always bombarded by UV light. The minute we go outside, we're bombarded by UV light. But we have this excision repair system, and it removes the damaged base pairs and replaces them. But if you don't have that repair system, then there's a likelihood of having a lot of mutations or even the increase of skin cancer. And this increases like 1,000 to 2,000 times because they can't repair their damaged DNA. Defects in genes required for DNA repair are frequently associated with cancer. So when you're missing these repair systems like proofreading, excision, nucleotide excision repair system, this really means any kind of those defects can mean the onset of having cancer. And this is the end of this chapter.